standardized around 24 techniques in diagnosing male infertility. And it's a pleasure for me to see my students. Dr. Srinivas is here, and Mrs. Shakuntala from Sharda Vilas, I could see her. Okay, so let me talk. It is just like an interactive session. I'll be dealing with male infertility, and any questions or any queries, please stop and interact with me. As you know, now we are facing a lot of that is a multifactorial disorder, especially 2024 is considered the World Health Organization. It has mentioned that India will be the capital for diabetes and obesity in the year 2024. And we are facing a lot of health issues. You must be knowing it is a multifactorial disorder, especially like diabetes, obesity, infertility, PCOD, thyroid issues, cardiovascular disorders, and cancer, different cancers, breast cancer, prostate cancer, many more. And you must be knowing World Health Organization, it has given a definition for a healthy individual. Health means it is not only the physical health, it is always focused on mental health, social health, and sexual health, as well as a spiritual health. The spiritual health is a new addition, and sexual health is very important for humans. And you must be knowing reproduction is the ultimate goal of all living organism. Any organism on this earth, from COVID virus till human, all living organism wants to contribute their genes to next generation. And today, based on that, I just focus on what are the causative agent for infertility. Infertility is an upcoming problem. Earlier days, couple infertility was very less, least. But in this decades, we have seen lot of infertile cases, especially in the IT sector, in youngsters. It could be due to the environmental factors, their diet, lifestyle could be one of the risk factor in causing male infertility. And today we'll discuss what are the causes for infertility. So the title is Unravel the Roots. What are the roots for male infertility? Generally, people, when they speak, they don't share much about the sexual health. Sexual health is also very important to be a healthy individual. And infertility, it is always defined as a failure to achieve a pregnancy after 1 year of regular unprotected intercourse with the same partner. This is the WHO definition. And why this is very important? Because nowadays there's an increase in the prevalence of male infertility. Globally, it is 60 to 80 million couples suffer from infertility every year, and 15 to 20 millions in India alone. We have huge number. And in the world population, the prevalence will be 0.74%, but this is not a small number. And what are the conditions? Generally, when we say an infertile individual, when we analyze the semen, so if the count of the spermatozoa in the semen, if it is less than 20 million per ml, then that condition is called as oligospermia. That means less sperm count. Then if the spermatozoa are completely absent in the semen, then it is called as azoospermia, no spermatozoa at all. Then it is a azoospermia. And if there is no motility in the spermatozoa, then it is called as asthenospermia. And sometimes the morphology of the spermatozoa will be totally different, and that is called as a teratospermia. They couldn't achieve the pregnancy because of the abnormal morphology. And sometimes we have a multiple condition, that is a low count with the abnormal morphology, count, less count, and morphology is different, that is oligoteratospermia. Similarly, low count with a low motility, they are not at all motile, then it is a oligoasthenospermia. And that is a OAT condition, is a low count, abnormal morphology, and low motility. So that is called as a OAT condition. This is really a very complicated thing, wherein there is a lot of problem. Count is less, then there is no motility, and also the morphology of the spermatozoa is totally different, thereby it is unable to achieve the pregnancy. And if you look into the etiology, what etiology means, the causative factors. What are the causative factor in causing male infertility? It could be anatomical. That means you must be knowing the sex organs like external and internal. The testis is the primary one, and we have accessory reproductive organs like epididymis, prostate, vas deferens, etc. So anatomical means if there is any problem in these structures, that is a primary or secondary accessory reproductive organs, then we consider that as a anatomical. Sometimes there is a formation of the tumor in the prostate or in the seminal vesicle or in the testes or in the vas deferens, or sometimes there is a blockage in the that is a vas deferens. Thereby there is no movement of the spermatozoa, and there is a calcification sometimes because of the infection. Infection is also sometimes one of the reason in causing infertility. And infection, if it moves, if it spreads to the internal reproductive organs, then in the testes or in the prostate or in the seminal vesicle. So those are the anatomical things. Sometimes in the, you must be knowing, there is a condition called as a CFTR mutation, cystic fibrosis. It is a genetic condition. Mutation in the CFTR gene causes cystic fibrosis. It's a genetic disorder where the person will be lacking this vas deferens. Vas deferens is completely absent in those individuals with the cystic fibrosis. These are all the anatomical. So similarly, we do have a that is a causative factors from the genetics part. That is, there must be Y microdeletion. You will be knowing, you must be knowing that there is a, in males there is a X and Y chromosome, and Y chromosome has a gene which are meant for spermatogenesis. And if there is any mutation happens in the Y chromosome, then it is a deletion, mutations or any deletions in the Y chromosome, then it's a genetic condition. Otherwise, if there is any changes in the number of the chromosomes, like in the sex chromosome, even then we can see there is a that in turn results into infertility. And endocrinological, if there is any variation in the hormone secretion or the level, like even testosterone, androgens, estrogen, some extent, and even TSH, all these things, maybe testosterone, then that is called as a endocrinological. Immunological, sometimes because of the breakage in the blood testis barrier, these spermatozoa will enter into the blood, and sometimes a torsion happens. These spermatozoa enters in the blood, and antibodies are raised against this, consider them as an antigen, and that causes a immunological problem. Thereby, spermatozoa will be killed. Sexually transmitted diseases, you must be knowing, this will be transmitted from the partners, and it could be there are a lot of sexually transmitted HTLV, HIV, and gonorrhea, syphilis. There are so many sexually transmitted diseases. These also causes infertility. Parasitic diseases, exposure to potentially toxic substance. You must be knowing, exposure to xenoestrogen. They act as estrogen, thereby they will inhibit the spermatogenesis. And very potent xenoestrogen is bisphenol. Bisphenol is rich in plastic. When plastic is heated, this will leach us into the water or the food item. Then when we consume, they act as a xenoestrogen, and thereby they are very toxins. And exposure to such toxins causes the damage or the deterioration of the these spermatogonial cells. And idiopathic, even we couldn't make out what could be the reason for this. So idiopathic generally having a Y microdeletions or mutation in some of the genes. Even sometimes not only the genes present on the sex chromosome, even the genes present on the autosomes are also very important. Mutation in autosomal gene, because the transcript of the autosomes might be very important for this reproduction. And always there is a coordination crosstalk between these autosomal genes, some of the genes, and Y chromosomal genes, and in the pathways, downstream, there are a lot of genes, they'll be activated depending upon the transcripts or the transcription factors released from the autosomes. So those are the idiopathic condition. Idiopathic means we can't make out what exactly the causative agent. They are called as the idiopathic condition. These are all the etiologies for male infertility. Why I focus on male infertility? When we look into the couple infertility, and males are more exposed to some of the things, and infertility is more in case of male when compared to females. In the couple infertility, 60% of the males have infertility when compared to females. So that's why etiologies of male infertility and knowledge about male infertility is very important, because you must be knowing ours is a male dominated society. Generally they don't accept that they are infertile, they have a less sperm count, they have a problem in the spermatogonial cells or the spermatozoa. That's the reason. So we are focusing mainly on the etiologies of the male infertility. So in this diagram, you can see impairment in normal spermatogenesis, that's the production of the spermatozoa. It could be due to a lot of other things. You heard about the varicocele. Even varicocele also always, because in the varicocele increases the temperature in the testes, thereby you must be knowing the testis is kept outside. It is a descended testis because the temperature of the testis will be lesser than 37 degree. It will be around 35, 2 degrees less than the body temperature. And because that temperature is optimal temperature for the spermatogenesis. So varicocele is an extra vein or the blood supply to that particular region, and that will enhances the temperature, thereby the spermatogenesis will be halted or impaired. So similarly, I told you environmental factors, estrogen in the environment, and testicular torsion, like a spinal cord injury also causes testicular torsion, and inflammatory infection, sometimes low grade inflammation also causes, that in turn results in immunological aspect, that is a like a autoimmune disorder. Body itself synthesizes antibodies against the spermatozoa. Similarly, testicular cancer, systemic diseases, hormonal dysfunction, testicular trauma, all these, retrograde ejaculation, anatomical. There is a blockage in the vas deferens, and thereby it won't reach. The spermatozoa will not reach, and it will be ejected out through urine. All these are things. There are blockages in different sites. So these are some of the reasons which will impair the normal spermatogenesis. And why we have to take up this study, why the studies on infertility is very important, even though there are people working on this infertility cases are there are reported, but no systematic studies have been done on the genetical, biochemical, endocrinological and immunological basis of male infertility. People might have taken either they are selected endocrinological or the genetic or some other immunological aspect, but holistically all round corner from all round, they have not approached this particular aspect. So present study help us to establish the etiology. So we could establish what is the etiology or a causative factor for male infertility in Indian population, in Indian context, and to understand the prevalence, what is the prevalence in India, and what are the type of anomalies which are more prevalent in our population, and degree of relatedness, how they are interconnected. And based on all these things, finally we can go for there is a better and precise method of diagnostic. So in our previous project, we try to develop some diagnostic markers, kits to diagnose male infertility. So these are all the things. So that's why the present study has been focused. And how to approach, how to address this question. First of all, in human genetics, you must be knowing, we should collect the information. That is, for that, we have to establish a genetic register, a genetic registry, including there we collect all the information, personal history and reproductive history, family history, all the information we'll collect in that. And pedigree. Pedigree is the study where we study different many generations, how the character has been inherited. That analysis will be done, chromosomal analysis, analysis of sperm sex chromosome aneuploidy through fluorescent in situ hybridization, and analysis of semen quality. That's also very important, gold standard diagnosis. And how the spermatozoa will function, the functional capacity of the spermatozoa is also very important. And nowadays you must be knowing oxidative stress is the culprit for main for all pathophysiological condition. Especially oxidative stress is the root cause for low grade inflammation also. So we want to evaluate the oxidative stress parameter in the semen as well as in the blood. And biochemical analysis of the semen plasma is also important. Immunological analysis, hormonal assays, molecular analysis, ultrasound scanning. So overall, so put together, if we do all this analysis, then only we'll come to know that what could be the reason for the infertility, because each person have a different issues. One person, if he has a hormonal problem, then another person will be having, there is a problem in the genetics, that is in the microdeletion, like that. So it is a multifactorial disorder, and all-round corner approach is very essential to find out the etiologies. So here, generally for any human genetic study, we have to recruit huge number of samples and equal number of control, normal individual and equal number of abnormal. Any disease, it could be infertility or diabetes or cardiovascular problem or any diseases, but same number of normal individual also, because it's a case control data set. So present study, I took a piece of work from the PhD from Dr. Srinivas. So where 250 infertile men and 250 healthy men, they were recruited from different hospitals and IVF centers in Mysore city. And in human genetic study, we have to take a consent letter from the participant. And before that, we have to take a ethical clearance from University of Mysore, Human Ethical Committee, and from IVF centers, from hospitals. We have to present our work and get the clearance from the hospitals to collect the blood sample and semen sample. And after that, the consent letter from the patient is also very essential to collect the blood and the semen sample for the analysis. And as I mentioned, establishment of genetic registry, very important document, because this genetic registry is used to collect the information like a family history. Just by looking into the family, we could make out what disease is more prevalent in that particular family, which helps us a lot. Some families, we can see there are more, that's some members suffering from cardiac problem or diabetes, many things, or infertility. So it is an alarming call for all other family members that compared to others, the onset of the disease will be in their early age when compared to others. So family history, medical history, then reproductive history, lifestyle factors, all this information will be collected through the questionnaire, and the interaction will be carried out with the infertile individual and with the family members to collect all the information. And the interaction will be carried out 2-3 times. And because first initially we have to develop a good rapport with them, because all these are very personal information, family history, medical history, all these things in one interaction, they never reveal the information. So a good rapport with the individuals, many interaction, once they are convinced, then because we are not supposed to reveal anything, and we have to maintain a lot of ethical issues are there here. So this is a establishment of genetic registry. After that, it is a, we do pedigree analysis. Pedigree is a diagrammatic representation of a family relationship, and it is a visual tool for documenting the inherited disorder. It's a very simplest tool where we can make out the pattern of inheritance of a disease, how it has been carried out from generation to generation. All this information we could collect from the pedigrees. It's a visual tool for documenting inherited diseases. And in our study, we found that genetic factors account 10 to 15% of severe male infertility, include chromosomal aberration and single gene mutation. And recent study shows that association between the rare sperm, genetic sperm defect with a diverse inheritance pattern, the family may be transmissible to the male. So this information is from the pedigrees. That's why pedigrees are very, very important. This is a representative pedigree, because for 250 cases and 250 control, we have to do the pedigrees like this. And these diagrams indicating this indicated the female, the circle indicated the female, the square indicated the male, and this diagonal line indicating they are dead. So this is the 1, 2, 3, 4 generation pedigree from both paternal side and from the maternal side. So by constructing this, we could make out whether there is any consanguineous marriage. Consanguineous marriage means marriage between the relatives. Even marriage between the relatives also also transmit some of the mutation in the family. All those information, 4 generation information, we could collect by constructing the pedigrees. These are all representative pedigree and representing infertility among the sibling. So these are indicating the infertility. This arrow mark indicating the infertility in this particular male. This indicate no child here. So this is indicating the infertility. So consanguineous marriage, even that in our study we found that 7% of infertile male, when we collect the family history, showed a positive association between a consanguineous marriage. Generally, not only infertility, this consanguineous marriage, as a genetist, I never entertained consanguineous marriage. Consanguineous marriage means marriage between relatives. Because what happened, for the expression of any genetic disorder, both the alleles must have undergone mutation. So we have more than 15,000 genetic disorders. Even then, the prevalence of these genetic disorders are less. The reason is always they need to be in 2 copies. Both the alleles have to undergone mutation. Then only it will be transmitted to children, and the children will be they'll be abnormal. So how it will happen? If they belong to same families, for example, uncle niece marriage or first cousin marriage in Muslims, or uncle niece marriage just in Hindus, what happened? Sister will be having the mutation, her brother will be having the mutation, and she will transmit the mutation to her daughter. And when her daughter marriage, uncle niece marriage, then what happened? Both male and female, they will be having the mutation, and if both the copies come together, then the children will develop the disease. That too in 3:1 ratio. That's why it is called as a Mendelian inheritance pattern or single gene disorder. So this is for the single gene disorder where consanguinity plays very important role. But whereas in case of multifactorial, like PCOD, male infertility or female infertility, thyroid issues, diabetes, obesity, cancer, not only the genetics, even the environment also plays very important role. That's why it is called as a multifactorial and polygenic. Not one gene, many genes are involved, and external factors also very important in causing this disease. It could be PCOD or male infertility or female infertility, anything it could be. So I told you, other than that, semen analysis is a gold standard diagnosis. Even though we do molecular analysis, before that, it has to be proved from the semen analysis. It is a very simplest one. Even then, it gives a lot of information. Thereby, we could narrow down our study. So instead of running around the bush, we could design the experiment targeting some of the genes depending upon the semen analysis. Semen analysis is one of the most important diagnostic tools to assess the fertility status of a male. So here we do the physical examination, very interesting. So always the body fluids, either that is a mucus or the semen or urine or fecal matter, they are the indicators of diseases. So if the urine color is yellowish, it indicates there is an infection. If the urine color is with red tinge, that indicates there is some internal capillary breakage. All these things. So urine and that is a fecal matter, semen, even sputum also indicates whether there is any infection or not. All this body secretion, they indicates whether the person is healthy or unhealthy. So physical examination, when we observe the semen, that is by observing the coagulation, liquefaction time, pH, color, viscosity, and volume, we could make out what exactly the problem lies. For example, here, yeah, coagulation. So semen, when it comes out after ejaculation, initially it will be in a liquid state. Semen is in a liquid state after ejaculation, but which gels shortly thereafter. After some times, it becomes like a gel. So if it won't, still if it is in the liquefied condition, if it won't gel, then absence of, that is called as a coagulation. Okay, absence of coagulation is indicative of congenital absence of vas deferens and seminal vesicle. So if there is no coagulation, immediately we could think that there is a congenital absence of vas deferens. It is a CFTR mutation, direct indication. We did not do any other test. So by observing the coagulation, immediately we can think there is a absence of vas deferens. Vas deferens is because of CFTR mutation. It is a condition called cystic fibrosis. For the confirmation, we can go for scanning. Okay, so that's the first one. Liquefaction, the gel state of the semen will liquefy between 20 to 30 minutes after that. So initially when it comes out, it is a liquid. After that, it will becomes gel. Then this gel once again liquefy after 20 to 30 minutes. And if it left at room temperature, and if there is any problem in liquefaction, this condition is called as a liquefaction. And if there is any problem in the liquefaction, it indicates there is a dysfunction of accessory glands such as a prostate. Nowadays, you must be knowing leading cause of death in men is a prostate cancer. Leading cause of death in female is a breast cancer. So it indicates there is some problem with the prostate gland. So it may be tumor in the prostate, infection in the prostate, prostate enlargement, or whatever may be, but there is something wrong with the prostate. So for the confirmation, we can go for scanning. So the third one is the odor. The normal semen has a distinct smell, which is like a pungent smell occasionally. So normal has a different odor. And if there is something wrong in the individual, then the odor will be different. A malodorous pungent smell is indicative of infection. The odor itself indicates there is an infection, while an odorless semen is associated with abnormal prostate function. Odorless is associated with the abnormal prostate function, and pungent smell is indicative of infection. So for the confirmation, we can take up other, that is a higher end investigation. Normally, it smells like a garlic or the clove, some spicy smell it smells in the normal individual. So these are all the physical examination, though it looks like simple, but it gives a lot of information. Thereby, we could narrow down our study. The color I told you, color of sputum, color of urine, color of fecus, color of semen indicates that whether the person is healthy or not. So most of the sample have a whitish opacity on ejaculation. So we'll collect the semen in a semen collecting jar, and we could make out whether it is normal or abnormal, because the normal semen has a whitish color. And if it is and after a liquefaction, they acquire a grayish or a translucent. And if the color is with a reddish tinge, then there is an internal breakage. A capillary breakage is there. Or if it is a yellowish, definitely there is an infection. Like that, we could identify what could be the reason. Similarly, the volume of the ejaculate should be measured and in a centrifuge tube. And low volume may indicate there's obstruction of reproductive tract. So if there is any obstruction, if there is a calcification or due to calcification, there is obstruction, then the volume of the semen will be less. And also congenital absence of seminal vesicle. If the seminal vesicle is absent, then even then we can see there is the volume of the semen will be low. Similarly, viscosity. This must be determined after liquefaction, high viscous. So it will be like a liquid, but if it is more viscous, it indicates you some infection. If it is a very thick thread like sometimes highly viscous, infection or absence of, or it indicates there is a anti-sperm antibody immunological problem, the presence of anti-sperm antibody, they are killing the spermatozoa. And then immediately what they do, if the semen is a very viscous, then it will be subjected for bacteriological and immunological assays investigations. Thereby they'll confirm whether there is a presence of anti-sperm antibody or any infection, and what could be the cause for the infection, all these things. So physical examination itself gives lot of information. And later on, after physical examination, we'll move on to microscopic examination. In the microscope, we can observe the density of the spermatozoa, amorphous particles in the spermatozoa, agglutination. Sometimes all of them, they agglutinate. They are not the normal spermatozoa. Count of the spermatozoa, vitality, morphology, motility, all these things. And in the motility, how they move, rapid linear, sluggish linear, all these things, non-motile. So different grades will be there. Based on that, we can say they are non-motile and motile spermatozoa. All these things we could decide. The shape of the spermatozoa is very, very important, because which reflect the normal development of the sperm in the testis, which can indicate normal spermatogenetic process. So male infertility is generally characterized by dysfunction process of the penetration, addition, and fusion of the spermatozoa with the oocyte. So it has to penetrate through the zona pellucida, and finally the DNA of the spermatozoa should reach, the nucleus should reach the ovum, and then the fusion happens. So after this, the physical examination, microscopic examination, we should know the functional capacity of the spermatozoa, not only the motility, whether functionally they are normal or not, even that's also very important, the functional capacity. So in order to confirm the functional capacity, we need to conduct a series of experiments to confirm. The first one is a hypoosmotic swelling test. Then second is a nuclear decondensation test and acrosomal intact test. These are the three gold standard test will be performed to know whether it has a normal function. This is the result of hypoosmotic swelling test. And here in the diagram, you can see this is the head of the spermatozoa. This is the tail of the spermatozoa in the microscopic examination. And this is the abnormal spermatozoa. These are all normal, because you can see there is a bend in the tail portion. So different kinds of bends you can see. Okay, so this is normal. And more than 60% normal, then we consider this as a normal. And why this test is important, hypoosmotic swelling test? This test is done to check the membrane intactness. You must be knowing spermatozoa is a single cell. Okay, so there is a cell membrane, then there is a nucleus, and during a spermatogenesis, during spermiogenesis, what happened? This will alter, it becomes specialized cell, and centrioles will move to the neck portion and it elongates. There's a quenching of the cytoplasm. Cytoplasm will be completely eliminated, and mitochondria will move to the neck region, and it supports like that. Okay, so Here, this test is it is done to check the membrane intactness. That's a plasma membrane permeability, whether the plasma membrane is permeable or not. In order to know that, this test will be done. The normal sperm will show tail bending, as I mentioned, due to increased hypotonicity, which break the disulfide bond in the tail fiber. Whereas in case if there is any damage in the plasma membrane, the intactness is lost, which decreases the osmotic swelling. Therefore, the tail remains straight without any bending. This in the previous diagram I showed, no, this is a abnormal which is not responding for the test because the plasma membrane integrity has been lost. And these are all the normal because the plasma membrane integrity is still maintained and they are responding for the test. And in the test, there is a breakdown of the disulfide bonds in the tail fiber. That's why they become curly. The curly are the normal sperm. And the tail of the spermatozoa, if it is straight, it is abnormal. So this is very, very important test to check the functional capacity of the spermatozoa. With this, though it looks simple, but this tells that whether the sperm is normal or abnormal. Second is a sperm nuclear decondensation test. So here, what happened, you must be knowing DNA is wrapped around histones. In the, I am talking about the somatic cells. So except the germinal cell, except spermatozoa, in our body, all the cells have there is a nucleus, and inside the nucleus, there is a DNA. And DNA is wrapped around histones. In spermatozoa, these histones are replaced by the protamines. These are also protein. Histones are also protein. Protamines are also protein. But in the spermatozoa, histones are replaced by protamines. In sperm, the somatic cell histones are. In the somatic cells, there are histones. DNA is wrapped around histone. You must be knowing the nucleosome and the solenoid structure, all those things. There's a different levels, degree, the DNA will be wound around, and thereby it will become so compact, and it could accommodate in a very tiny chromosomes. That is in the somatic cell. But in the spermatozoa, the histones are replaced by protamines. And these protamines are small positively charged testis-specific nuclear protein that facilitate chromatin condensation and compaction. So in order to have a compact of the DNA in the spermatozoa, high condensation, high degree of condensation, and more compactness, these protamines are very, very important because they are small positively charged particle. DNA is negatively charged. So there is that makes a super compact of the DNA in the spermatozoa. There is a reason why there is a replacement of histones by the protamine in spermatozoa. The reason is, which gives lot of protection of sperm from the DNA damage. What happened in our body if the DNA is damaged due to any mutagen or due to carcinogen? Then what happened? The damage is confined to the individual. It will not transmit to next generation. If the damages happens in the spermatozoa or oogonia, then it will be transmitted to next generation. So that's why this is the beauty of the nature. So every level, there is some defense mechanism. So nature designed the system in such a way that if something goes wrong with the somatic cell, it is confined to the individual. If something goes wrong with the germinal cell, it will pass on to next generation. So more precaution or more defense mechanism has been inculcated in the reproductive structures, reproductive organs, or in the these spermatogonia or oogonial reproductive cells. So more protection of the sperm from the DNA damage, and also to synchronize the cell cycle between the sperm and the oocyte, and also to increases the hydrodynamicity of the sperm. For all these purpose, the protamines, all the histones are replaced by the protamines. But what happened? This will be in the spermatozoa. During fertilization, once again, the protamines are replaced by histone. It has to be. It cannot have a protamines always. So this is a temporary arrangement, and this is only in the spermatozoa. And after the fusion of the spermatozoa with the oogonia or the oocyte, then what happened? Once again, there is a replacement of the protamine by histones. That has to happen. If it won't happen, then it is an abnormal spermatozoa. And the third test, you must be knowing acrosome. Acrosome is like a cap-like structure in the spermatozoa, and 3 by 4th of the spermatozoa having this acrosome. Acrosome has more than 40 hydrolytic enzyme. And this is a test. You can see there is a hollow nature. This is the spermatozoa which shows a positive response to this acrosomal intactness test. These enzymes are involved in the capacitation. The acrosome is involved in the capacitation and chromatin decondensation, acrosomal reaction, all these things. So, as I told you, this acrosome portion has more than 40 enzymes called as acrosomal enzymes, and they're all hydrolytic enzymes. They play an important role in penetrating the spermatozoa into the oocyte. Very, very important. Otherwise, even though the spermatozoa, even though it will reaches the oocyte, it will not penetrate. It will not break the zona pellucida and enters into the egg. That is the thing. So acrosome enzymes are very, very important in many infertile condition. If the spermatozoa doesn't have this acrosomal enzyme, if it is lacking acrosomal enzyme, they are unable, they will reach the cell, but they are unable to penetrate into the oocyte. So it is a proteolytic in nature. It will break the proteins in the zona pellucida and thereby it could penetrate. And acrosin is a trypsin-like serine protease that is exclusively found in the acrosome of mammalian spermatozoa. Acrosin is the major enzyme. It is a trypsin-like serine protease, so which will break down the zona pellucida and present in the acrosome of mammalian spermatozoa. It's all associated with the zona pellucida binding, and easily it penetrates the into the that is the egg or the oogonia or oocyte. So these are the tests indicating that there's a positive association. And nowadays you must be knowing, and in our body, during a metabolism, body will release some kind of toxins that is called as a reactive oxygen species, and in vivo, in vitro. So if you look into the source of reactive oxygen species, both in vivo and in vitro, we can see this reactive oxygen species. One mechanism is during metabolism, this mitochondria will release some extent of reactive oxygen species. Apart from outside, also through our diet and some of the lifestyle, we are getting a lot of these reactive oxygen species or reactive nitrogen species. So together they cause oxidative stress. You must heard about the oxidative stress. Oxidative stress is associated with all kind of health issue. For anything, they correlate with oxidative stress, not only the physical health, even mental health is also associated with the oxidative stress. So oxidative stress is a condition established due to an imbalance between antioxidant level and reactive oxygen species. So more of reactive oxygen species, reactive nitrogen species, hydrogen species, then together it enhances or it causes oxidative stress. But always I used to tell everyone, human body is a mesmerizing thing. Always know, we quote a lot about, we appreciate about 7 wonders or 8 wonders in the world. Thinking about China Wall, Taj Mahal, so many monuments, thinking they are all mesmerizing, amazing. But human body itself is a mesmerizing thing. If you look into each mechanism at molecular level, really you will be astonished. And all molecular aspect mechanisms are designed in such a way that perfection to the core and accuracy is 100%. Nowhere, there is no scope for any errors, even in DNA replication or transcription, translation, unimaginable level accuracy you could see. Nowhere any error. If error happens, finally you must be knowing that leads to some diseases or pathological condition. So even in cancer, onset of cancer, it won't happen in the early age. It is associated with the age, because accumulation of lot many mutation has to happen to develop the cancer. There must be a failure in all level. Every level there is a defense mechanism to causes cancer, uncontrolled proliferation of cells, and de-differentiation of the cell. All these things because there is a failure of all defense mechanism. There must be failure in the DNA replication, then fidelity in DNA replication, DNA repair mechanism, mutation in the tumor suppressor gene, mutation in the proto-oncogene, all these things, failure, then finally end result is the cancerous condition or uncontrolled cell proliferation. So similarly, always in our body, even though there's oxidative stress, yeah, and internally lot of antioxidant enzymes are present. So there are many enzymes which try to buffer this oxidative stress inside, and even we could supplement this antioxidant from outside. That's why all vegetables and fruits, they are rich in antioxidants. Even some berries, fruits, so rich in this antioxidant supply, definitely it will boost the immunity. So here there are many enzymes inside our body that will nullify this reactive oxygen species. And what are the causes for this oxidative stress? It could be hobbies like a smoking or exposure to high temperature, pollution. It could be any kind of pollution, toxic chemicals, endocrine disruptor. I told you this bisphenol is an endocrine disruptor. Always there is a competition for bisphenol for this testosterone receptor. So it is an analog, and thereby it will bind to the testosterone receptor. Thereby testosterone cannot bind to that receptor, and that is an endocrine disruptor. And these are all some of the factors which causes oxidative stress. This is oxidative stress, I told you, and this is could be from the oxygen species, nitrogen species, hydrogen species, etc. And it is might be from the drugs or smoking, pollution, and prolonged state of spermatozoa present in the epididymis. All these things. Finally, this causes a protein damage. Lipid peroxidation increases. Biomembrane damage. I told you, plasma membrane permeability is lost. It is because of the damage of the plasma membrane. Could be oxidative stress. DNA damage. Ultimately, the spermatozoa will be damaged, and end result is infertility. So we need to evaluate the antioxidant capacity, and to protect the spermatozoa from oxidative stress, seminal plasma is endowed with a numerous enzymatic antioxidant. So when we compare the seminal plasma with the blood plasma, the seminal plasma has more this antioxidant than the blood plasma. So here it has in vivo, it has a superoxide dismutase, an enzyme which will kill the superoxide ions. Catalase is another antioxidant enzyme, and non-enzymatic antioxidants are also there. That is from the food derived, vitamin C, vitamin A, vitamin E. These are all antioxidant. I told you, food derived means it is the vegetables or the fruits or berries. They are rich in all these antioxidants. So when we compare the control with all different kinds of infertile condition, we can see there is a superoxide dismutase SOD enzyme level is high in the control when compared to infertile condition. Lipid peroxidation is more in this infertile condition when compared to control. Similarly, reactive oxygen species level is very high in infertile individual when compared to control. This graph represent that this oxidative stress is more in case of infertile individual when compared to control. Could be one of the risk factor in causing male infertility. And it shows that increase RYS in the different infertile condition, that is teratozospermia, oligoasthenospermia group, where motility is observed, and they are less motile when compared to normal. And apart from that, this is one part. I told you we need to have an all-round approach for this because it's a multifactorial condition. Lot many reasons are there in causing infertility, and each case is a unique case. Each person, the infertility in each individual will be different because it could be due to biochemical aspect or endocrinological or genetical or from the external environment, all these things. So we need to diagnose from all around corner. There's a biochemical analysis of the seminal plasma. The accessory gland. So we have a primary reproductive organ, that's a testis. Accessory reproductive organs are epididymis, where the maturation of the spermatozoa happens. Then seminal vesicle, prostate, vas deferens, all these are considered as an accessory reproductive organ. Not only the testes or the epididymis, even all these accessory glands are also, health of accessory glands are also very important. The accessory gland produces substance specific for each gland, which can be used as a marker to determine the glandular function. Each one could act as a marker in diagnosing the infertility. Secretory function of the seminal vesicle is reflected by fructose. You must be knowing the spermatozoa has to travel a lot from where it has been originated. It has to reach the female, that is a uterus, or it has to move into the fallopian tube. So this, it is a very lengthy journey. It requires a lot of fuel for that, energy for that, and that is supplied from the seminal vesicle. That seminal vesicle, always it reflects by the fructose. And the importance of the fructose estimation is mainly in case of azoospermia, as there is absence of fructose indicating a congenital absence of seminal vesicle. If there is no seminal vesicle, then there is no fructose. So no fructose, there is no fuel for the spermatozoa to reach. So similarly, citric acid and acid phosphatase are the reliable indicators or the biomarker for the prostatic function. Nowadays, you must be knowing after some 60 years or the 55 to 60 years, men are is very susceptible to develop prostate enlargement and calcification in the prostate, tumor in the prostate. Prostate cancer is very common, leading cause of death in men. So citric acid and acid phosphatase are the reliable indicator of health of the prostate, and their presence is in low concentration or their absence indicative prostate dysfunction. Low concentration or absence indicating there is something wrong with the prostate. Then for the confirmation, we need to do some scanning and other higher end diagnosis for the confirmation. So even here in our study also, we found that there is some alteration in the seminal fructose distribution among different infertile group, and seminal citric acid, that is a distribution also varies. So fructose, I told you, very important. It will break down and it converts into energy, and this energy supports the motility of the spermatozoa, and that's why the fructose or the function of the seminal vesicle is very, very important. And similarly, citric acid also, it indicates, I told you, the health of the prostate. Okay, so reduced seminal citric acid, it could be indicating there's an inflammation or a chronic prostatitis. That indicates, then they have to look into whether the prostate is normal or healthy or unhealthy. So because low grade inflammation also indicate the variation in the reduced seminal citric acid, and chronic prostatitis also could be identified. And complete obstruction of ejaculatory duct also we could make out from the citric acid, reduced seminal citric acid. And other than that, endocrinology or the hormone profile is very, very important. One of the major factor in male infertility, and it is less than 2% generally. So here, always we consider three hormones. One is a testosterone and LH, FSH. Luteinizing hormone, follicular stimulating hormone. These are the enzymes. Always this hormone level, this is the hormone profile, normal in the abnormal individual. So if you look into the etiology, if the person is suffering from primary testicular disorder, then there is an increase in LH LH and FSH, and there is a reduced testosterone. And if these three hormones are reduced, then it indicates hypogonadotropic hypogonadism condition. And testosterone is normal, LH is normal, there is an increase in FSH. This indicates seminiferous tubule failure or Sertoli cell only syndrome, all this indication. And testosterone normal, increase, LH increase, FSH normal, androgen resistant syndrome. Then all three normal, disorder of sperm transport. So like that, based on this chart, this is a chart which tells us that there is a, depending upon the hormone profile, how we can decide the etiology of the male infertility. And we could see there is a decreased and increased level of this FSH, LH, and testosterone in different infertile condition. And distribution of hormone profile with respect to clinical condition, definitely we can see there is a significant differences exist among this. And next we have to move on to the chromosomal analysis, the distribution of infertile group analyzed for chromosomal analysis. And here the numerical chromosomal abnormality, we could see around 4 cases, structural chromosomal abnormality, Y chromosomal abnormality. All these things are quite common in male infertility. We have to rule out all this, whether it is due to hormonal, whether it is due to chromosomal variation, whether it is due to microdeletion. Like that, we have to find out and rule out the condition. And finally, based on the study, finally we could develop a definite diagnostic marker, or we could design the treatment, how it could be treated based on the condition. Here, we can see all these different conditions. There are conditions like a Klinefelters. They are also infertile. And where they have a XY 47 chromosomes, mosaic pattern. There is a XY, some of the cells, and some of the cells, XYY. That's called as a mosaic. Means when we observe 100 cells, some cells are normal and some cells are abnormal. So that condition is called as a mosaicism. And depending upon the percentage of the mosaicism, the clinical or the phenotypic feature is decided mainly based on the percentage of the mosaicism. And some of them are XYY. And structural variation also we can see in the chromosome. There's a translocation, there's a deletion, and addition part of the chromosome. Yes. Huh? Addition in the Y chromosome, and sometimes Y chromosome is very small, very tiny. Thereby we can see there is a lot of microdeletions, inversion in the Y chromosome, etc. So this is how they look like. This is the elongated Y. This is addition in the Y chromosome. This is inverted. D is a inverted. E is a small due to microdeletion. These are all the karyotypes. You must be knowing these are human chromosomes. Zoology and genetics people, they generally do karyotype. But for others, this is a human chromosomes. It is called as a karyotyping. Sequential arrangement of the chromosome is called as a karyotyping. And we have 23 pairs, that is a 46 chromosome. 44 are autosomes, and 1 pair, it is a 6 chromosome. In males, 1 X and 1 Y. In female, this X will be 2 in number. So here, there's an addition, extra part from another chromosome as added here. That's why it is a abnormal. Here, there's a translocation. Chromosome from 6 to 17. There is a from other chromosome, a part of the chromosome has been translocated here. This is translocation. Similarly, very tiny Y chromosome here. And inversion, it has been inverted. Here it has been inverted, arrow mark indicating that. XYY, 2 Ys are there. And aneuploidy through fluorescent in situ hybridization, we could make out sometimes there is a single spermatozoa has XY, and when it fuses with a X bearing X, then it will be XXY Klinefelters, like that. Okay, apart from this chromosomal abnormality, when we look into the microdeletions, then Y chromosome harbors several genes responsible for normal fertility, and testis-specific gene responsible for normal quantitative and qualitative spermatogenesis are, there are series of genes RBMY1, DAS, CDY, and these are ubiquitously expressed. They are responsible for the development of secondary characters, enhancing the reproductive function, and certain housekeeping genes are there for the normal function, USP9Y and PCDH11Y, all this. So this is the Y chromosome. This Y chromosome has been divided into AZFA, AZFB, and AZFC. These region has genes. These genes are very, very important for the production of spermatozoa. And this is a TDF, testis determining factor. And SRY gene is also there. This is the SRY gene, which will determine the sex of an individual. Y chromosome, this SRY gene, this is present. This is called a pseudo-autosomal region. Both the sides, it's a pseudo-autosomal region. Only this region is involved in the recombination. Rest of the things are not involved in recombination. And if SRY gene, by chance, if it involved in recombination, then we can see there is a condition called sex reversal. Genotypically XY, they are males. Phenotypically, they are females. Similarly, genotypically, they are XX, but phenotypically, they are males. It is because it is called as a sex reversal because of the translocation of SRY gene. So this is a gel profile showing a PCR amplification of DAS gene. Here, the infertile individual see here, complete absence of the DAS gene. Similarly, the marker SY255, this is SY254 in the DAS gene absence, and here it is absent, SY255 in different individual. This is from the different infertile individual. Some of them, they have these microdeletions. Some of them, they don't have microdeletions. This is for the DAS. Similarly for the RBMY1 gene. So there is a mutation in some of them, gel profile showing the PCR amplification. This is for the Y microdeletion for different regions like AZFA, AZFB, C. So in the C region, we can see 58.3% of the Y microdeletion. And out of 180 infertile individual, 50 control individual, 12 infertile individuals show Y microdeletion. So that especially in the together, it is C region, AZFC region has 58.3% of Y microdeletion. So after that, even when we look into the ultrasound scan of the male infertile individual, we can see a lot of anatomical changes, and this will be done through color Doppler, that's ultrasound scan, and transrectal ultrasound scan. So testicular volume is also very important. Assessment of testicular volume, whether what is the size of the testis in relation to spermiogram. Spermiogram means, already I mentioned, all the things, that's a physical and microscopy, functional, those are all the spermiogram. And here, in correlation with that, we are assessing the testicular volume, the internal organs, primary organ and accessory organ, to confirm the infertile condition in comparison with the fertile or the normal individual, to check whether there is any variation. Testicular volume was significantly lower, not only in infertile men, but also in elderly. Aged people also, we can see there is a regression in the size of the testes. And also in infertile individual, there is a degeneration or regression in the size of the testis when compared to normal individual. This is scanning of normal testis. And this is a hypoplastic. Hypoplastic means there is a reduction in the size of the testis. So this we could identify in infertile individual, not only infertile, in aged one. If they are 60 years, 70 years, then gradually the size of the testis is reduced. That is called as a hypoplastic testis. This is a scanning indicating that there is a calcification. I told you, infection, if it is not treated, if it is neglected, sometimes the urinary tract infection, if it is not treated, then it will enter into the testis region, then moves to epididymis, then moves to seminal vesicle, prostate, and entire internal reproductive organ will be with infection. And more infection causes the calcification. These are all the calcification internally in the testis. This is the right testis. Lot of calcification, too much of calcification. So always infection we shouldn't neglect. Even dental health is also important. If they don't have not hygiene, then many bacteria, they enter into the from through oral cavity, they'll finally migrate to the testis, and finally they will degenerate or the causes infection in the testicular, the seminiferous tubules in the testis. There is a significant differences between the normal and abnormal or the infertile individual. Similarly, hydrocele. Hydrocele is abnormal collection of fluid between the parietal and visceral layer of the tunica genus, an imbalance between the fluid production. If there is a fluid production, fluid accumulation of the fluid, that is called as a hydrocele. Hydrocele is the most common cause of painful scrotal swelling. Scrotum will be swollen, and this also causes infertility. So this is hydrocele of the testis. This is observed through ultrasound scanning. Hydrocele in both, this is in the right testis, enlarged and fluid filled cavity. The scrotum will be swollen. Similarly, varicocele. Varicocele is an abnormal dilation and elongation of the internal spermatic vein and pampiniform plexus of the spermatic cord. So because of this varicocele, spermatogenesis will be inhibited, and about 15% of the men have problem due to this varicocele. It could be operated. Sometimes even there is a recurrence. After some times again they will develop this varicocele. So this is the varicocele. Even varicocele causes infertility because of more temperature. Varicocele increases the temperature, and increased temperature reduces the production of sperm, oligospermia. And this is the epididymis, and there is atrophy in the epididymis. There is a regression of the epididymis. Epididymis is very important, where maturation of the spermatozoa happens. And cyst, huge cyst present in the epididymis, it also causes the infertility. And after this, until then it is a scanning. Then later on, for the internal prostate, vas deferens, seminal vesicle, they do transrectal ultrasound scan. Until then it is a color Doppler. This is the prostate. This measurement of the prostate, and in our study we can see hypoplastic, reduced size of prostate. This the size of the prostate is reduced, and there is a huge cyst present in the prostate cyst. As person ages, prostate cyst and prostate enlargement is very common problem. Sometimes infection in the prostatitis or tumor in the prostate, if it is neglected, that causes prostate tumor. That's a cancer and a leading cause of death. This is the prostate cyst, huge cyst here in the prostate, which causes infertility. And this is a calcification. All these white patches are an infection in the prostate. This calcification also causes, that is because of the infection. Severe infection causes the calcification. This is the prostatitis. Complete prostate is abnormal now. It is a prostatitis. So we can see there's a significant differences between healthy individual with the infertile individual with reference to the prostate. This is the first report related to the prostate abnormality in the infertile individual. Yes. Yeah, this is the seminal vesicle, and even there is a cyst in the seminal vesicle. All these are the cyst in the seminal vesicle. Seminal vesicle, I told you, it will secrete the fructose, very important for the survival of the spermatozoa and also the motility of the spermatozoa. Hyperplastic, size of the seminal vesicle is very small. This is with all these ranges we have to calculate. Hyperplastic, enlarged the seminal vesicle. Any organ enlarged or regressed, both are pathological condition. So ultrasound scan, all these things, it is a non-invasive method, very easy to perform, provides diagnosis without any side effect. And this should be taken as a first order of investigation in male infertility. So infertility is a multifactorial condition. With one approach, it is not possible to find out the causative agent. So we have to have a multi-dimensional that is a approach from all corner. We have to approach, then only we could make out what could be the reason for the male infertility. And this is how the infertile cases will be diagnosed from the research point of view, how exactly we have to carry out the research on male infertility. So with this, hope it's a time. Any questions? I'm happy to interact with you.
Ma'am, good evening.
Yeah.
Ma'am, I have one question which is not related to male reproductive system. It is you said that in infection is there in urinary tract. So if there is UTI, then this leads to infertility in male. So does this happen even in female?
Yeah, absolutely. Pelvic inflammation, that's why we have to be very much, we have to give more focus on the hygiene. What happen no, if we neglect, if there is unhygienic condition, especially when we go to public toilets or during periods, if we are not maintaining hygienic thing, then the infection will gets inside, which causes pelvic inflammatory disorder, and in turn that will uh, causes infection in the fallopian tube, uterus, and one of the reason for female infertility also.
Thank you, ma'am.
All this data I took it from Srinivas thesis and from Dr. Sharad Kumar. So
Srinivas sir is my colleague.
he's a participant here, because I thought of giving a talk about human genetics. I thought you are all from life science and you know more about all the things. So I thought let me take up this.
Yeah, ma'am. Thank you so much.
All the credit goes to Dr. Srinivas. He has standardized all the techniques and he has published around 34 papers. And I'm very proud of him always.
Madam, I have one question.
Yeah.
SRY gene present only in the Y chromosome.
Yes.